As we look at what's happening under different conditions, we are looking at hormonal regulation, and the two big hormonal molecules that are involved with our metabolism is insulin and glucagon. We looked at insulin last time, and now we're going to look at glucagon. Glucagon is the signaling molecule that is secreted in higher concentration when we're under a low energy state or a starvation state. And so, if we look just back at one slide, insulin goes and affects a lot of different things: muscle, brain, liver, and adipose tissue. But when I go to glucagon, we only affect adipose tissue and liver. The reason is the muscle and the brain do not have receptors. And so if it doesn't have a receptor, it's not going to cause any effect. But in terms of liver and adipose tissue, we're going to create a situation in which we are going to flood the blood system with energy molecules. Adipose tissue is pretty easy to think about. Okay, the hormone-sensitive lipase is the lipase that breaks down the triacylglycerols, and this particular enzyme actually is involved with associating the lipase to the lipid molecules, the actual droplets, and so they work in conjunction to start breaking down the fats, and then the fat is exported, in this case as free fats in the serum albumin. So this, if the fat exports, is in free fatty acids. And then they're in the serum albumin carried by that protein. All right, when we hit liver, this is the one tissue that really controls a lot of things. Overall, glucose export, glycogen degradation, and gluconeogenesis. It affects these particular enzymes. All right, so can anybody tell me which pathway is this enzyme, which is being upregulated, affects? It is two pathways, glycolysis and gluconeogenesis. What happens to glycolysis with this pathway? And glycolysis, it's inhibited, and gluconeogenesis, it's increased. All right, what does this enzyme affect and do? Phosphorylase kinase? It what? It adds a phosphate to the enzyme glycogen phosphorylase, and glycogen phosphorylase does what? Breaks down glycogen. Okay, and so that is the molecule that upregulates the breakdown of glycogen. All right. Obviously, you know what this one does, right? Glycogen synthase, and it turns that one off. Okay. Can you tell me what enzyme does? Protein phosphatase 1. It does take off a phosphate from, not a protein. It is a protein that takes off a phosphate. But what phosphate does it take off? Okay. Oh my goodness. I know you know this. All right. It takes off the phosphate off of glucose 6-phosphate. When you remove the phosphate of glucose 6-phosphate, you get glucose. And what can happen to that glucose then? It can actually be released. If it's phosphorylated, it's sequestered inside the cells. So this is what allows the glucose to be released. All right. And that can be coming from gluconeogenesis and or glycogen degradation. And then the phosphofructokinase, version 1. What pathway are we talking about? Glycolysis. And that reinforces glycolysis from occurring. Okay? or sending the glucose out into the bloodstream as we start into starvation. All right. So, knowing what these enzymes do and why they do it upon response of the glycogen might be an important piece that you need to know. All right, there is one more slower acting regulator that affects tissue types. And these are the PPARs, or peroxisome proliferating activated receptors. Now, the peroxisome proliferating has absolutely nothing to do with what they do now. It's just they were found in enzymes that had peroxisomes active. We now know that these are regulatory events for metabolism. They just kept the old name. Most likely at some sort of review in the next few years, they'll all be reclassified and renamed to actually name them a little bit better what they actually do, and so it'll confuse everybody. There are different versions of these, alpha, beta, gamma. I think there's also a delta somewhere. But what they do is this. They detect free fats or fat derivatives. So free fats that get inside the cell or derivatives of fats that build up in the cell, and they will bind to them. Now, the fats in the cell, if you have a very tiny amount, that's normal. But if you have a large amount, like in the case of starvation, then they will get up into a high enough concentration to bind one of these PPAR receptors. Now, they usually bind through a binding protein, and they come in and bind to the PPAR receptor, which then dimerizes to another protein. And when you get this complex, this one, and that one all together, they become a transcription factor, which then will produce genes that will then start utilizing lipids as energy sources. Okay, they'll also, in different tissue types, do a whole bunch of other things. Give you kind of an overview. They affect liver, muscle, and adipose tissue, and depending upon which version, alpha, beta, delta, you can get all sorts of things. But in general, if you have lots of fats in the liver, you start oxidizing them, breaking them down. You start to have so that you can convert into ketone bodies. You can also synthesize lipids. You can also upregulate the pentose phosphate pathway. In skeletal muscle, you have fatty acid oxidation. And in adipose tissue, you can have lipid synthesis and adipose differentiation under some of the signaling molecules. And under other signaling molecules, you can have fatty acid oxidation. And so it's just a different pathway that is much more slow acting, and they work in conjunction with insulin and glucagon. All right. When you do take a look at starvation, what you start to think about is, okay, where are we going to get the energy to do all of this? And so there are lots of different energy molecules in the body. And so I give you two different tables that are in two different books. They are similar, but not exactly the same. All right? And so depending upon what you're thinking about, your typical 70 kilogram person and what types of energy sources there are. In this is just the idealized case. You only have 15 kilograms of fat adipose tissue. That's great. Some of us have a little bit less, some of us have a little bit more, but the fats, if you take a look at both of these cases, regardless of just slight differentiations, over 100,000 calories. And the reason why we use calories is because most nutritional labels are still in calories. Followed by that is the protein. You've got protein next. That is misleading. The amount of energy that you have in protein both here and down here, they call it muscle, okay? Misleading because that would be all of your protein, okay, your musculature, and you can't utilize all of that. You can only utilize a portion of that effectively. Then you've got glycogen that's in the skeletal muscle, which is a larger source. Glycogen in the liver, which is a much smaller source but more dense. And then you have the mobile energy in the circulatory system. And you can see if we used 1,700 calories, okay, if we actually did a conversion about all of this energy, you can survive starvation numerically about 100 days, right? Not really, okay? That's if you utilize every single amount of the molecules, and most people's metabolism cannot do that. But if you take a look at it, 83 days is represented by fat, 14 days in muscle, glycogen in the skeletal muscle, a little over 8 hours, the liver 4 hours, and the circulatory system represents about an hour and a half worth of energy source. And so we can go, there are some extreme cases, but when you get out to 40 days of fasting, that is extreme, and you get closer to death than life at that moment in time. All right. So how in the world do we utilize all of this? In starvation, we change the metabolism, okay? And we're looking at the liver cell in particular because that's the most, that's the tissue type that responds to starvation conditions and changes its function quite a bit. So what in the world happens? Okay, under starvation, we've got to keep the brain going. So this is where the key is. Oxaloacetate, the citric acid cycle, runs up through gluconeogenesis. Okay, and so we take a lot of oxaloacetate out of the cycle. And as you can see, there's a dotted line for your citric acid cycle. Basically, the citric acid cycle at the top stops. Okay, but you're signaling the rest of your body to say, hey, I'm under starvation conditions. Where is most of the energy? Most of the energy is in the fat. So you are starting to break down fat. You're mobilizing it. You're sending it to the cells. And the muscle can use it. The brain cannot. The liver can take up that fat that's being mobilized. Well, what in the world happens? Well, as you remember, fatty acid oxidation, we make lots of acetyl-CoA. And to make lots and lots of ATP, I need to react acetyl-CoA with oxaloacetate and create citrate. But the problem is I have very little oxaloacetate that is available to react with acetyl-CoA. So what the liver does, the concentration of the acetyl-CoA goes up and creates the ketone bodies. And ketone bodies are exported. Now, what happens at that moment in time, the concentration of molecules in the bloodstream, the ketone bodies become much higher. The muscles can use ketone bodies, and under these kind of extreme conditions, the brain can start using it. Now, that ketone bodies production is always going on because the heart is always using it, and so ketone bodies are actually happening. All right, the other source of energy that you have is in protein. All right, and we will take the amino acid and we'll take the amine, make lots of urea, so the waste product goes. But then what happens to all of the carbon chains, or if you remember, a lot of the carbon chains in the amino acids enter in the citric acid cycle. And they can be converted and end up as oxaloacetate. And that oxaloacetate will then go up and export glucose. So a lot of our energy sources to keep the brain alive come from proteins after prolonged starvation. And because we will use up our glycogen stores pretty quickly. back out here, okay, all glycogen in and of itself, if used directly and completely, give us not even a whole day's worth of energy. Okay, it can be depleted very, very quickly. What's important is to make sure that you have enough blood glucose concentration to keep your brain active, and they cannot go below about 4.4 millimolar. It can't go below 2.2 millimolar. The normal is about 4.4. And so we actually try to keep that concentration up. Here is a good graph of what happens. All right, day 0. We tend not to have much ketone bodies because really the heart is only using it, so it's not very high in concentration. We do have a basal level of fat that is always circulating, okay, and that is normal. And we usually have a pretty high concentration of glucose that we try to keep it at, okay, and these are in millimolars. If you think about it in other terms, that's 90 to 100 milligrams per deciliter. As you continue starvation over long periods of time, so 10 days, 20 days, 30 days, what is the dramatic change? Ketone bodies are a huge change, going from almost non-detectable to very, very high. What else do you see that goes down? Glucose. It does dramatically drop in a very short period of time, but then steadies out. That's the level that we want to keep it at a minimal level so that your brain actually functions well. What do you see of fats? They do increase, but they don't increase a whole bunch, do they? Okay? Why in the world do the fatty acids not increase that much? You do start to release them, okay? But what happens is they are being sopped up by the liver and converted into ketone bodies very, very quickly. And so fat doesn't roam around as much because the liver is overproducing that ketone body. And so it rises a little bit and it continues to rise, but ketone bodies are very, very high. Now, the muscle can use fats, so that's fine. Muscle can take this in and can use it. Muscles can also use ketone bodies, that's fine. Glucose exclusively for the brain, but then the brain can also take on ketone bodies and it'll start working. And so you can see that this dramatic shift, really in the first 10 days, you've got lots of ketone bodies. All right. What do you think, let's say, starvation over periods of time? Some people use starvation or partial fasting, they call it, as a diet technique, right? And they will fast, not in any spiritual sense, but they'll use it to diet. What do you see as the benefit here? You can lose fat pretty quickly in those conditions because you start converting all of those fatty acids into ketone bodies, which then can be used. And so there are other consequences of that, okay? But periodic fasting can actually break down fats pretty quickly. Now, fuel use also has something to do with how sustained your exercise can be. And so if you take a look at the rate at which you can use energy in terms of ATP, okay? ATP, there's not a lot of it running around, okay? Only 223 millimoles in your entire body, in your muscle. The fastest way to reconvert ADP back to the triphosphate is phosphocreatine. It does over 70 millimoles per second, but there's not a lot of capacity. So it's short term in terms of how much exercise you can do. We can increase this by taking creatine, right, and increasing the amount there. If you convert muscle glycogen into lactate, that's anaerobic exercise. It is quick, 39. And you can extract a bit of energy from it. But if you take the glycogen, break it down into individual glucose, break it down through glycolysis, make it pyruvate, send it into the citric acid cycle, go all the way through the citric acid cycle, making the NADH and FADH2s, and then do the proton gradient and the ATP pumps, or the synthase. Look how much energy you can extract out of that glycogen. This is anaerobic. This is aerobic. Okay? There is a significant difference in how much energy you can extract. But look at the rate. Much, much slower. All right? Conversion of liver glycogen. Okay, so you got to think about that. Okay, I have the reserve in my liver. I have to go through glycogen breakdown. I have to cleave off the phosphate. I have to put it into the circulatory system. I have to make it go through the circulatory system, take it into the muscle cell. Then glycolysis, citric acid cycle, oxidative phosphorylation, and I extract energy. Yes?
So when you take creatine phosphate, how does it enter into does it go directly to the muscle?
It will be absorbed by all tissue. Muscle is the largest tissue type, and you're going to absorb a little bit over the entire. Most of it will be flushed out, though. But it's just by sheer mass action, you make the concentration so high in the bloodstream that it will leach into the cells. Yes, it's beneficial because you can have very intense workout for a longer period of time. Very intense, very intense, very intense. Not as intense, right? And so this is the amount of energy that you can use that is very intense. And so if you just have more of the creatine, you have larger amounts of this, more intense workout very quickly. Yes?
So going back to the slide before the ketone bodies,
Mm-hmm.
starvation state.
Yep.
Like I know that like ketone bodies make a lot of acid you can also get into ketoacidosis.
Mm-hmm.
So is that a problem that over a long period?
Yes, it is a problem over long periods of time because you do get that acidic conditions in your bloodstream, which then can have things misfunction because it's in the wrong pH scale.
So, like I know you're saying that it can cross the blood-brain barrier for the brain.
Mm-hmm. The ketone bodies can.
So what kind of effect would that have on the brain?
It is not. What you do is you get a little bit lethargic and you don't think as well. Take something as, for example, I don't know if you've ever watched the show Survivor, right? They make them go through those competitions, and some of the competitions are really quite simple. But if you've been under starvation conditions without lots of sleep, you can see the effect of them not being able to think as straight. And so even though you're under survival mode and your brain is functioning and you can have that repolarization event because you have energy, doesn't mean that you're working efficiently. And so your cognitive ability actually goes down over that period of time too. Now, if you convert fats from the tissue into CO2, check out how much energy is available there. That is a ton of energy, but the rate at which it is being able to extract is much slower. The amount of intense activity, or how hard you can go, is directly linked to how fast you can extract energy. I gave you a little outline. Here's a better outline about until fully depleted under extreme or intense workout. Muscle, 3 seconds, 6 seconds, 3 minutes, and this is intense exercise. Liver glycogen, 50, muscle glycogen, 1.5, and then about 5 hours. If you are being chased, all right, like for real, you're running really fast, but you slow down over time. Now you push yourself, you can continue to go and go and go, but just because you have the will and the mindset to do it, you will run out of energy sources. and your body will break down and stop. It isn't necessarily just mind over matter. There is a limit to what you can do. It can be seen most efficiently or directly in terms of a runner. Okay, a 100-meter sprinter can sprint that 100 meters in under 10 seconds. Okay, under 10 seconds, what kind of energies are you using? ATP, the phosphocreatine, you're not really burning, maybe a little bit of glucose, right? But you're using these very, very quick stores. The rate at which you can produce this stuff is very, very quick. You start going further, all right, the velocity or the intensity in itself goes down because you have to shift into use of other slower methods by which to extract energy. By the time you get up to a marathon, which someone actually did break the 2-hour barrier recently, that is just wild. And unfortunately, it wasn't official, so there wasn't an official record, but actually it was a pretty big barrier that stood for a very long time. You can't extract the amount of energy fast enough to really change that time very much. All right? There are tricks for marathoners to try to increase their rate. Okay? Most of the time, way back when in runs, there were people that stood by the run and they would hand out cups of what? What would they hand out cups of if you've ever been a runner? Water. Okay, yeah, you probably need some water, right? Because you're sweating, you're trying to get that back. Now, they don't just give out water. What people do is they will, under extreme races, they will get gel packs of carbohydrates. Why? So that they can get those very fast or faster sources of energy so that you can have a faster rate, extended rates, or extend the amount of fast rate extraction of energy. Have you ever worked out, if you've ever ran, you get to a point, you're running, you're getting tired, you're like, oh, I'm just going to stop. But what if you go just a little bit longer? What happens? You get a runner's high. Some other people talk about second wind. Some people have never run their entire life. Okay? And you get to a point where you're thinking, oh, well, I can continue to do this. All right? What happens is you actually, during your workout, change your metabolism. You actually shift from those fast energy stores like sugars. Runners sometimes that are going to do long runs, carbo load the night before so that they have higher concentrations of sugar in their bloodstream. But eating once for a very long run doesn't actually benefit you over the long run. But you shift, and by the time a marathoner is finishing the run, almost no utilization of glucose. Okay? What they've done is they've shifted over to a very efficient use of about 50% protein and 50% fat. Okay? The time at which you do that changeover, okay, to utilize different energy sources and how fast you can use those energy sources is really part of your conditioning. Okay? It's not just getting tone. It's not increasing the strength of your muscles as the only benefit of being well fit. It's actually allowing your cells to upregulate the metabolic pathways faster than someone who doesn't do exercise. And so that's part of it. Also, you get more muscle mass, so you get more ability to use those pathways better. All right. The other thing we're going to talk about is signaling. Okay, there are other types of signaling that we're going to talk about and how hormones work a little bit. We are a very highly controlled entity, and most of our control comes from the brain, nerve impulses. But we take those nerve impulses and convert them into molecules that affect tissue types and affect what happens inside of those tissues. And so there are a couple of pathways. One is neuronal. It's a signal between the neurons, and it's actually a very short distance control. Hormonal is kind of what we use classically when we talk about all of these things, and that's probably more vernacular. But hormonal is long distance signaling. So a small molecule is released to control something, and it controls over long distances. Most of the hormones are whole body influencers, and they're relatively slow. And so we've got long distances and short distance. The actions and the results are very, very similar. It just depends upon how close and what signaling molecules are actually happening. Hormonal control is through receptors, and so a lot of these are signal transduction pathways. We talked a lot about the receptors that are in the plasma membrane where the hormone binds, and usually we've even used H as the hormone, and then you bind a receptor and you have a signal transduction. Usually they're very fast. Second messengers are created, you get amplification, and you can alter the cell's function very, very rapidly. Other hormones that usually are very hydrophobic will actually bind to receptors that are internal to the cell, sometimes all the way inside the nucleus to become transcription factors, and their regulation is upregulating the DNA to produce new proteins. That usually is a longer acting, takes a longer time. When we have hormones, there are a couple of releases we can have. An endocrine, which is released into the whole body. That's the kind of one that we probably default and think about. There's a pancrean that's released into the extracellular spaces, but that affects only neighboring cells, very short distance. And then we have autocrine, where a cell will release a molecule and it will bind to itself to affect itself. So endocrine, pancrean, and autocrine. Some of the hormone classes that we've been talking about: insulin and glucagon, small peptides, somewhere between 3 and 200 amino acids. Normally, you have one peptide that becomes one hormone, but sometimes the peptide is synthesized a little bit large. It is cleaved into several pieces, and you get active hormones and several hormones. Insulin is released as a longer peptide, cleaved in two different places to get what is called the mature insulin, which is the one that's more effective. And so some of these have to be processed. The catecholamine compounds start from a parent, and so all the way up, phenylalanine can be converted into tyrosine into dopa or L-dopa, which is a signaling molecule, a neuronal signaling molecule, into dopamine, and then norepinephrine and epinephrine. And so there are a whole class of signaling molecules coming from amino acids. We've talked about the eicosanoids derived from arachidonic acid, and those will affect the prostaglandins, thromboxanes, and leukotrienes. So we've talked about those. Those are part of the fat hormones. Steroids, classic ones are the sex hormones, and they are derivatives of cholesterol, so there's a whole class of those. Vitamin D is a hormone that regulates calcium in the blood. The retinoids regulate cell growth, survival, and differentiation. They are derived from beta-carotene, and they are related to the retinal that we use in our eye. Thyroid-containing compounds are kind of interesting. They are small organic compounds that are full of iodine. And so very important molecules, and we'll see why in a little bit. But iodine is not necessarily something that we think about eating very often, and the government has helped us, okay, and placed a source of iodine in our body so that we can get that source. Does anybody know where the government regulates and mandates that we have iodine placed into a food source? In salt. Okay, and so they're iodinated salt. Why in the world? Okay, you don't need a lot of iodine, but iodine, once it gets in the body, really goes only to one tissue, and that's the thyroid. And so in that tissue, iodine really is very, very concentrated. This is good because most of the iodine containing hormones have at least 2, 3, or 4 iodines that are bound to it. It's kind of unique. It also creates a liability in terms of if there was any nuclear accident and or war, because a lot of the radioactive material that is gamma radiators that are very, very toxic to us are based upon iodine. And so if you're exposed to radioactivity of that kind, then it gets into your body. And since it isn't distributed all across the body, it goes right to the thyroid and concentrates it very, very concentrated with all of that radioactivity, and it leads to cancers, and in a very, very bad place. Way back in the 60s, when if you went to elementary school, you had a fire drill and you also had a bomb drill. What did they do? They hide under the desk. Like, that's going to do anything, okay? Made themselves crazy, but at that point in time, in the bomb shelters that everybody was making way back then, you put water, you put food, and you put anti-radiation pills in your bunker. Do you know what the anti-radiation pill was? It was it was potassium iodide. Okay, and it was to protect you against if you're exposed to radioactive iodide. Then if you overcompensate by having a lot of clean iodide, then by competition, you'll excrete more of the radioactivity than it be concentrated into your thyroid. It was anti-radiation for one single type of radiation. And so, yes, it had some benefit, but it was mostly placebo. In that, oh, the bomb went on. I'm going to take this potassium iodide and I will be all okay. But that was the idea. Another signaling molecule is proteins that produce nitric oxide, NO. NO is a gas, and this is kind of an interesting signaling molecule. The NO is fairly hydrophobic and can diffuse through membranes. And so it is a gas that is signaling molecule. I actually used to give an article, a journal article, and had people look up a activator of a nitrogen oxide producing substance that caused vasodilation. And after they read the full article, they realized, oh, the drug was Viagra, okay? And Viagra works through a mechanism that is a nitric oxide gas regulator. It is stimulated by guanylate cyclase, which is analogous to the adenylate cyclase enzymes. So a whole class that are just gas molecules. Okay. Regulator of the regulators. Okay, how does the brain send out signals? Well, there are certain tissues that send out hormones that affect everybody, affect the whole body. And 2 of those that you can think about are, for males, testes, the ovaries for the females. But there are other tissues. The kidneys, the pancreas, the adrenal glands are all going to give off molecules that are hormone regulators. The problem is the brain doesn't affect these directly, okay? The brain sends a signal and it's going to work through 2 different targets, the hypothalamus and the pituitary gland, and send it out to the thyroid, the adrenal, the pancreas, and all these. And so there is an order by which these signals are released. And so we get sensory input from the environment, and that stimulates our brain to do something. Many of these are not conscious regulators. They're in our unconscious mind that actually these signals go through. And what happens is the nervous system will send actual electrical signals to the hypothalamus, which is the first for almost all regulation, and the hypothalamus then will release a small molecule. So it goes from a neuronal to a molecular signal. And that molecular signal will be released in the bloodstream, and then it will hit its first target, which is the pituitary gland. Now, the pituitary gland has two parts, the anterior and the posterior, the one in the front and the one in the back. And from there, the pituitary glands will send out small molecules. And you can see that most of those small molecules are proteins. Why in the world do you know that they are proteins? Well, if you take a look at the MR, and it says 4,500 or MR 28,000, what we're talking about is the molecular weight of that protein. Okay, and they get released. At that moment in time, those are released into the bloodstream, and then they will hit their secondary targets. And so things that you think classically, the adrenal cortex, thyroid, ovaries, testes, and then you start seeing molecules that you probably have seen before: testosterone, progesterone, estrogen. You may have even heard of cortisol. These kinds of molecules are things that you've heard before. And you only see that when we get down to this level. Okay? And that's because of the relative concentrations. Now, each one of those molecules will then pick a target of tissues to cause different effects. There are a couple of exceptions. The central nervous systems, the adrenal medulla here, kind of cuts out the middleman and produces epinephrine. Why in the world do we want the middleman cut out and we're going to give out epinephrine? We need that quickly, and so it is much more directly linked. The other place where we see lots of regulatory things that we understand, insulin and glucagon, not done by the not done by the brain, it is direct of the blood glucose level in the pancreas, and so sending out those molecules that we know. Why don't we know the other regulators above, let's say, the level that is kind of right here? Here's the problem, okay? The central nervous system, depending upon whatever the stimuli is, from physical pain to fear, okay, will signal the hypothalamus, and the hypothalamus will give out a hormone in a concentration of, what is in? Nanogram. Okay. Now, what is the concentration that you would have if you placed a nanogram of material in 6 liters of blood? It's got to be ridiculous in terms of its concentration, ridiculously small. Okay. Now, that's okay, because look what's happening. The hypothalamus releases a little bit of molecule, and where does it go? To the pituitary. So they're right next to each other. Only a very small concentration is needed, but this makes it a challenge for us. How do we detect that? For a long time, we didn't even know it was there because we can't detect that low concentrations. We're still identifying hormones and regulatory. Now, once the pituitary gets into it, look, it releases it at the microgram level. A microgram in 6 liters is still low in concentration, but we start to get to things that maybe we can detect. It will then hit the secondary target, and we get concentrations now in the milligram level. Okay? And so difference between here is 1,000-fold. A difference between here is 1,000-fold. So a difference between here is a 1,000,000-fold increase in the molecule. At this point in time, these are the ones that we can detect and have been able to detect for a long time. So we knew about these These types we're still trying to find out because they are released in such small concentrations. All right? And so there is an element of a signal transduction pathway in terms of expansion, or you get a much larger response from a small response. And so it's mirrored here in the hormones release.